So this summer I worked at the University of Michigan with Dr. Anirudh Hirve, and pretty much I worked on trying to find a cure for brain cancer. What we're trying to do is find a new chemotherapy option that will work with the already existing ones. We realized that in brain cancer cells there's a protein called PDI that's overexpressed, which just means that it's extremely abundant in those cells. So we are trying to find something that will inhibit it or block it from either being created or from functioning properly. So we already have found a few compounds that do inhibit it, which is such as PACMA or estradiol, and the problems with those is that they bind forever, so those can have long-term side effects, and we're trying to find a novel drug that will bind and be beneficial to our bodies, etc. So what I did was I screened over 100 compounds, and I was looking to see if they bound to PDI and where they bound, because where they bound would decide if it could bind and then come off, or how it would affect the body.
Over the summer, I worked at the University of Michigan in the SEN lab in the intern health study. This study looks at the effects of stress, genetics, and environment on depression rates of first-year medical interns, so their first year out of medical school when they've been matched up with a residency program. This is a time that's very stressful. It has long hours, inconsistent hours, lack of sleep, people work holidays, and compared to about 5% of the general population who's self-reporting as depressed. After this intern year, 25 to 40% are self-reporting as depressed. So the benefits of having this known time of stress is that usually in mental health studies it's hard to find cause-effect relationships. So stress causing depression is depression causing stress. But with this, you can measure the interns before this year and throughout and after, and see actually how this known stress affects them going forward. The specific work I will be doing is looking at educational trajectory, so the ranking of their medical school versus the reputation of their residency program, and seeing if an upward trajectory maybe gives positive feedback and momentum to get through those stressful times and actually lowers depression rate compared to those who are going from a higher ranked medical school to a lower ranked residency program and might be getting less positive feedback and having more of a lack of confidence going into the year, and so they feel those factors more heavily and are more likely to become depressed. That research we haven't run the analyses for that research, but that is our hypothesis, and we'll see what the results show.
So this summer I worked at the University of Michigan in the biomedical engineering department, and I worked with mouse ovarian follicles and growing them outside of the body. What I did personally was I cultured the follicles in a 2D environment and in 3D where they were encapsulated in a hydrogel to see how they grew, which way they grew better, and if the hydrogel would work to be a good environment for them to grow in outside of the body. The end goal of the whole thing is to find a way to grow human ovaries outside of the body for, especially for kids who had cancer and whose reproductive organs were damaged by chemotherapy, so that they could still go through puberty in a more natural way rather than standard hormone injections and other things like that.
There you go. Suicide is a commonly researched topic. There are a lot of studies done on suicide and depression, or the link between suicide and depression, or the link between suicide and sexual orientation. But there aren't as many studies being done on suicide and body image. So this past summer, I worked at the University of Toledo, and we took responses from the YRBS survey. It's a national survey that's given to middle schools and high schools across the country. And we took questions that were specifically related to their perceived weight status. So they would respond if they thought that their weight, regardless of their actual weight, if they thought they were slightly obese, slightly underweight, just normal, obese, underweight, and the suicide questions. So have you ever considered suicide? Have you ever been seriously injured in a suicide attempt? Have you ever seen a doctor? And so using the statistical software SPSS, I conducted a secondary data set analysis, which is just a fancy way of saying we didn't collect the data, someone else did, and we are trying to find a link between suicide ideation and body image. That's it.
did my research on Davenport Schnitzel sequences, which are sequences which are subject to constraints under parameters n and s. The three rules are that 1, they can have at most n distinct symbols in them. 2, no two elements in the sequence can be the same consecutive ones. And 3, if you take any two symbols in the sequence, you can look at just the occurrences of those two symbols A and B, and then you look at you take out the repetitions of A and B immediately, so you get a sequence of just A, B, A, B, A, B. That sequence can have lengths at most s plus 2. So the question is, what is the maximum possible length of such a sequence? This problem has been pretty much solved for constant s or constant n. So and then you take the other parameter and you let it grow to infinity. So my question is, what if, for example, s is approximately the square root of n, and then you let n grow to infinity? What is the behavior of the length of the function? So it was known to be on the order of n squared and n to the 5 halves, and I showed, among other things, that it's exactly n to the 5 halves.
This summer I worked in a U of M research lab. It was for a translational oncology program. So basically what I did is I did a PDI assay, which is an assay, just kind of like a fancy word for experiment, and I screened a series of roughly 500 compounds. So with this, I was trying to find a drug that would inhibit a certain protein that is overexpressed in a certain type of cancer. It's called glioblastoma, and it's found mainly in the head and neck of like the older population. So my results were kind of very straightforward. I found 17 active compounds. An active compound, by our definition, was a compound that had an IC50 value of above 40%. So what an IC50 value is when the compound inhibits half of the protein that we're trying to stop. And so with 40%, that would mean that it was inhibiting a majority of this protein, so it would suppress the cancer. So the drug that we're hoping to create, we're hoping it will be able to be reversible, because my lab has already discovered a drug called PACMA31, which does inhibit this protein, so it already suppresses the cancer. But however, it's not reversible, and the long-term effects are not great on the human body. So we're trying to find a drug that is reversible, therefore we won't have to have those long-term effects. So my lab is still working with the drugs that I found. I found roughly 17 active compounds, and so they're going to put them into a mouse trial and then later add them to a clinical trial depending on what drugs were the best.
Over summer I worked at Ford Motor Company within the Human Factors and Ergonomics department. We tried to make cars more comfortable by looking at the egress and ingress, which basically just means going out and going in of a vehicle. And then we filmed the ingress and egress using the same type of video recording equipment that they use for some animated movies and video games.
This summer, I worked at a lab at Michigan State University in the College of Veterinary Medicine, and the lab was looking at a cell signaling pathway called Notch signaling, and Notch signaling determines what type of cell a stem cell may become. And in Notch signaling, you have a sending cell and a receiving cell. And on the sending cell, there are two ligands that I looked at called jagged and delta-like. And then when those ligands touch Notch receptors on the receiving cell, that'll send the signal. It's like, oh, now it's time to be a bone cell. And so in my research, I looked at how jagged 1, jagged 2, delta-like 4, and delta-like 10 sent signals to become bone cells. And then I also looked at 3 enzymes called fringes, and I looked at manic, radical, and lunatic fringes to see how they promoted or inhibited the pathway to make it more efficient or less efficient. And my results are a little inconclusive right now. So the lab is running a couple more tests with my samples that I was using, and we're just going to see where that takes us.
This summer I worked at U of M at the School of Education with Dr. Bain and the rest of the Big History team. So what they do is they look at the course Big History and they make sure that everything is going smoothly, and they come up with new ideas for the course. So Big History, for those of you who don't know, or I guess just for you, is a class that's kind of new and up and coming, and it's being taught at Green Hills right now, and it's about science and history, and it's not really a normal history class, but it's more like history of the universe. And so what I did was ran data through a statistics program called SPSS, And what we did was we looked for models and linear regressions that correlated different variables such as age, gender, religion, school type, and area code against scores in written essays. So what I would do is look at correlations between gender and scores in the mechanical section of an essay. And then from there, we are hoping to make changes necessary to make sure that everyone is kind of being educated in a similar way and to the best of the teacher's ability, and to make sure that everyone's getting the best scores possible.
This summer, I worked at the University of Michigan using the National Survey of American Life Adolescent Sample to look at the effect of parent-adolescent relationships on adolescent anxiety within African American and Black Caribbean adolescent populations, as well as to see if the parent-adolescent relationship could be moderated by age, gender, and ethnicity. We used SPSS to analyze the data, and then we used STATA to weigh it so that it was nationally applicable. And what we found was that parent-adolescent relationships are not in fact moderated by age, gender, or ethnicity, but it does have a direct effect on adolescent anxiety. But that was for the upweighted data. Within the unweighted data, we actually found that there were correlations between the moderating variables and parent-adolescent relationships. So we explained that due to the small sample size.
Over the summer, I worked in an HIV lab at University of Michigan. Imagine HIV like this. This is the envelope, and this is the virus inside. Envelope helps the virus get into the cell, kind of like this. Two things can happen. Either there can be an active infection where this virus uses the cell's mechanisms to create more virus, and there are antiviral drugs and the body's own immune system that can hinder this. However, a latent infection can also occur, in which when the virus can actually insert its own genome into the host cells. Though there's no more virus actively being created, anytime it can disengage from the host genome and more virus can be created. And that's a source of persistent infection. My lab hypothesized that a certain type of blood cell stem cell could be a reservoir for this persistent infection. They hypothesized that the way we tested this was we looked for identical sequences, important sequences of the viral genome in the blood stem cells, and we compared them to HIV sequences in mature immune cells. I found an amino acid match in a donor in one of the helper T cells to one of the existing stem cells. And if there can be more of a connection made between the two, if there can be more matches made, then this could prompt further research in targeting those stem cells as sources of persistent infection, which could lead to a cure.
So I was also at the University of Michigan, and I was in the chemical engineering building, and I had a couple tasks to do. I was going in to help out with the 3D printers with some grad students who are designing and printing valves and other types of devices. And first I had to solve a problem that they were having where the printer was printing things too small compared to what they designed. And that could be a problem when the things you're printing are on a small scale from 50 to 200 microns even. And after I got the size sorted out of the prints by adjusting the settings little by little, seeing how much better the print would get, I was able to design and print the valves that they wanted me to. And they were just something called pneumatic valves, which basically just means that they're controlled by the thing that controls whether or not it's open or closed is basically just air pressure. And the printer that I was working with was their newest one and their most expensive and complex. So it could use 5 different materials, well more, but 5 at a time. And I was trying to get it so that we could print a flexible membrane in the same print as the standard plastic that you print the valve with.
I spent time this summer working with Dr. Gattapalli at Mott's Children's Hospital at the University of Michigan. We were researching the way that pediatric patients make decisions when dealing with fulminant ulcerative colitis, and whether or not, like what factors went into their decision between having surgery and having medicinal treatment.
Over the summer, I worked at a vascular lab at the University of Michigan, and we looked at the effects of e-cigarettes on the cardiovascular system, and specifically the abdominal aorta, and its relationship to a disease called abdominal aortic aneurysm. And we looked at e-vapor without nicotine, cigarette smoke extract, and then we had a control. And we found that e-cigarettes and cigarette smoke extract had a significant effect on AAA formation and the abdominal aorta.
Interesting.
Alright, so this summer I worked at the University of Michigan Transportation Research Institute, and I worked on assessing the potential correlations between driver attributes such as age, weight, and gender, and how they use their armrests during transit. It was actually sponsored by a global automotive interiors manufacturer called Yanfeng, and they supply interiors to like Porsche and BMW and Ford, I think. So yeah, it was spurred by that, and the data was actually taken from a previously completed study.
This summer I worked in the computational neurolinguistics lab under Dr. Brennan at U of M. My project was diurnal and weekly variances in the auditory event-related potential, which basically means I looked at the brain's response to an auditory signal, over how that changes over the course of the day, like morning versus evening, and over the course of the week, so Monday through Friday. And what I found was that there was like a larger amplitude means a higher alertness in the subjects. So I found that there was a higher alertness in the afternoon, and I found that as the week went on there was a lowering of alertness. So subjects got more tired as the week went on, except for Friday, which had the highest alertness. We were looking at U of M students, so these are students who are excited for the last day of the week and are like more awake on that last day.
Cool. What was that?
During the summer, I worked at Ford Motor Company's Research and Development campus on their virtual driver systems team. And the team's goal was to develop an autonomous vehicle, which is a vehicle that can drive on existing roadways with little to no human interaction. On the team, I was primarily responsible for creating an engineering specifications list for each of the sensors on the vehicle, which enable the vehicle to sense the environment and deliver information to the software for the vehicle to make decisions on how to drive the car.
Over the summer, I worked in a lab at the University of Michigan conducting research on the aspiration-expectation gap in African American adolescents. So educational aspirations refer to the amount of education an individual would ideally achieve, whereas their expectations refer to the amount of education they believe they will actually be able to. And it often occurs that educational expectations are significantly lower than their aspirations, given that they call on the individual to think realistically rather than ideally. So the first question I attempted to answer was, is there a gap between African American adolescents' aspirations and expectations? And we found that yes, there was. But this has been shown in previous research to be detrimental to human progress, or to educational progress. So I wanted to see what variables were associated with the gap so that we can hopefully narrow it in lots of students. So the variables that I looked at were gender, GPA, and parent racial socialization. Parent racial socialization refers to the transmission of views on race and ethnicity from parents to children.
For my research, I worked with the U.S. Geological Survey to examine samples that they've taken of zooplankton in the Detroit and St. Clair rivers. And I counted species and measured them and looked for correlations between the species distribution and where in the river they were.